This is your Dr. Piri. Welcome back to lectures in endocrinology. Today we are going to discuss the function of the pancreas, the endocrine function of the pancreas. If you remember very well, we looked at the exocrine function of the pancreas, where we saw the production of enzymes that are involved in digestion of a lot of nutrients that you are eating each day. But here now we'll be biased towards the production of hormones because we are discussing the endocrine function of the pancreas. So there are a number of hormones that are being produced by the pancreas, the major ones being insulin and glucagon, which are the most common hormones that are produced by the pancreas. So this lecture is going to be biased towards insulin. There are lots and lots of metabolic function of insulin, so you need to understand a lot of metabolic functions of insulin. I know most of this information you might have covered it in biochemistry, but in physiology you really need to appreciate the function of insulin because there are a lot of pathology that is associated with insulin, especially diabetes. It's a very common disease, so you really need to understand the function of insulin. So you don't need to get tired. There will be a lot of information, but the more the information the better, because as a student you'll be able to understand. So at your own free time, take your time and just enjoy the information concerning the insulin. Okay, so this is lecture 8 of the endocrine system. We are going to discuss insulin, glucagon, and diabetes mellitus. Diabetes mellitus. So in this lecture mainly we are going to discuss insulin. So we'll have the general information concerning the production and secretion of these hormones by the pancreas. Here's the pancreas. It has got a function in the GIT because it's an accessory organ to the GIT. So you realize to say we have acinar cells that are producing enzymes that will be transported by the duct system. Then with the help of pancreatic duct to be released into the duodenum via the sphincter of Oddi. So that we discuss in GIT. Now I'm interested in the islets of Langerhans, where you have the endocrine cells, the glandular cells that are producing a number of hormones. So you have the alpha cells, the beta cells. The beta cells are the ones that are producing insulin. The alpha cells are producing glucagon. The beta cells that are producing insulin, they are also producing amylin. On top of that, we have the delta cells that are producing somatostatin. Then we have the F cells or the PP cells that are producing pancreatic polypeptide. So those are some of the hormones that are being produced by the pancreas. So there are quite a number of hormones. So let's start. The pancreas. The pancreas, in addition to its digestive functions, the pancreas secretes two important hormones, the insulin and glucagon, that are crucial for normal regulation of glucose. So you know the normal regulation of glucose, lipids, protein metabolism is actually regulated by insulin and glucagon. These are the two antagonistic hormones that are produced by the pancreas. The other minor hormones whose function are not well established include the amylin, somatostatin, and pancreatic polypeptide. By these hormones, they can also regulate the secretion of the other hormones, especially insulin and glucagon. So they can inhibit the production of insulin, okay? Especially the amylin that is also being produced by the beta cells. They can somehow inhibit the production of insulin, so they will regulate the production of insulin. Somatostatin, of course, can also inhibit the production of insulin and glucagon. Okay, but the major function of these hormones is not yet known. So you as a scientist, maybe you discover the function of some of these hormones. The physiological anatomy of the pancreas. The pancreas is composed of two major types of tissues. We have the acinar cells, which secrete digestive juices into the duodenum. They are secreting enzymes, so you have the duct system to transport the enzyme. And you notice so those enzymes will be activated by the enterokinase. So you have pancreatic trypsin that can be activated by the enterokinase. So it's being produced as trypsinogen. If you remember, all those are enzymes. So we discussed that. But now I'm interested in the islets of Langerhans, which produce insulin, glucagon direct into the blood. So we'll look at the regulation by which these hormones are being secreted as well. The islets contain three major types of cells. We have the alpha cells, the beta cells, the delta cells, which are distinguished from one another by their morphological and staining characteristics. So they'll pick up different stains and they have different morphology. So these are the three major types that are found in the islets of Langerhans: the alpha, the beta, and the delta cells. The beta cells constituting about 60% of all the cells of the islets. So it will constitute about 60% of all the cells of the islets, the beta cells. They lie mainly in the middle of each islet and secrete insulin and amylin. So they are the ones that are producing insulin and amylin, like I mentioned. The amylin is a hormone that is often secreted in parallel with the insulin, although its function is unknown. So the major function of amylin is not known, but we know to say to some extent it inhibits the secretion of insulin. Then the alpha cells, about 25% of the total pancreas or the total islets of Langerhans, they are producing glucagon. So 25% of the total islets of Langerhans, they are producing glucagon. So they'll be producing glucagon. Then we have the delta cells, about 10% of the total islets of Langerhans, and these are producing somatostatin. Somatostatin is a hormone that can inhibit the secretion of insulin. It's the same hormone that was also inhibiting the release of growth hormone. So somatostatin can inhibit the release of growth hormone as well. In addition, at least one other type of cell, the PP cells, which are also referred to as the F cells, they are present in small numbers in the islets and they produce a hormone of uncertain function, and this hormone is called pancreatic polypeptide. Pancreatic polypeptide, the PP. So the cells that are producing PP hormone, they are called PP cells or the F cells. The close interactions among these cell types in the islets of Langerhans allow cell-to-cell communication and direct control of secretion of some of the hormones by the other hormones. So there is control of hormone secretion by the other hormones. For instance, insulin inhibits glucagon secretion. The amylin that is being secreted in parallel with insulin is going to inhibit insulin secretion. The somatostatin is going to inhibit the secretion of both insulin and glucagon. So if you're not paying attention to this statement, I can bring a lot of MCQs there just from this statement. So you need to understand that there is interaction of hormones that can inhibit the release of other hormones. That's why the pancreas is releasing quite a number of hormones, not just glucagon and insulin. This is the anatomical structure of the pancreas, the islets of Langerhans. So we have different type of cells. The F cells or the PP cells are the ones that are producing pancreatic polypeptide. The alpha cells are producing glucagon. The delta cells are producing somatostatin. The beta cells, which are the majority, 60% of the islets of Langerhans, are the beta cells that are producing insulin, and they are also producing amylin. Okay. This is basically the same information. You have islets of the Langerhans and the different types of cells, the delta cells, the alpha cells, beta cells, and the F cells or the PP cells, which are not shown here. So there is rich blood supply to the islets of Langerhans. So you can see the capillaries there, because once the hormones are produced, they need to be secreted. That will be transported by the cardiovascular system. That's why you have these blood vessels. The insulin is a hormone associated with energy abundancy. So now we are going into the metabolism of insulin, or the function of insulin. We'll be biased towards insulin. Then the other lecture will discuss glucagon. Insulin is a hormone associated with energy abundancy. So after eating a meal, after having your meal, you will know to say that the body, the pancreas, will release a lot of insulin because you have high levels of energy. So insulin is a hormone associated with energy abundancy. That is, when there is a great abundance of energy-giving foods in the diet, especially the excess amounts of carbohydrates, insulin is secreted in greater quantities. So if you eat a lot of carbohydrates, you just know that there will be a lot of insulin that will be secreted by the beta cells. In turn, the insulin plays an important role in storing the excess energy. So you need to store the excess energy. So in the case of excess carbohydrates, it causes them to be stored as glycogen mainly in the liver and muscle cells. So most of that glucose that will be internalized by the liver cells, the hepatocytes, it will be converted into glycogen and stored within those cells. But in adipose tissue, it will be converted into fat, like triglycerides, fatty acids, and so on and so forth. Also, all the excess carbohydrates that cannot be stored as glycogen are converted under the stimulus of insulin into fat and stored in adipose tissue. So some of these glucose that cannot be converted into glycogen, it will be converted into fat and stored within the adipose tissue. In the case of protein, insulin has a direct effect in promoting amino acids uptake by cells and conversion of these amino acids into proteins. So you can see here that insulin has got a lot of functions. So it's causing the storage of glucose into glycogen in muscle cell and hepatocytes. It's causing the conversion of some of the glucose into fat and stored within adipose tissue. Is also causing the conversion of glucose into amino acids, and some of the amino acids can be internalized by the cells in the presence of insulin. So the same insulin that is stimulating cells to internalize glucose is also stimulating certain cells to internalize amino acids that will be converted into proteins and being stored. So you can realize to say when you eat high carbohydrate food, the chances of you gaining is going to be more, because most of these glucose will be converted into fat, it will be converted into proteins, it will be converted into glycogen and stored within the cells. And then the size of the cells will be increasing, the adipose tissue will also be increasing, and then you start gaining. That's why if you're drinking a lot of alcohol, alcohol contains a lot of glucose or a lot of sugar that can be converted into fat. If you're drinking a lot of drinks, especially carbonated drinks or just any kind of drinks, they contain a lot of glucose, they contain a lot of sugar, all that will be converted into fat. That's why you start gaining. Even people who drink a lot of opaque beer, I'm talking of Chiwuku here, so most of you know Chiwuku. So if you're drinking Chiwuku, you start gaining. Why? It's because most of that glucose will be stored in form of glycogen, fatty acids or fat in adipose tissue, and also protein in other cells. In addition, it inhibits the breakdown of proteins that are already in the cells. So if you have a lot of insulin, the same insulin that is stimulating the cells to internalize amino acids to be converted into proteins, it will also inhibit those cells to break down proteins that is already stored in the cells. So you're not going to be using a lot of proteins in the presence of insulin. The insulin chemistry and synthesis, the production of insulin and the structure of insulin hormone. The insulin is a small protein. Human insulin has a molecular weight of 5,808 kilodalton. This is the molecular weight of insulin. It is composed of two amino acid chains connected to each other by disulfide linkages. So you have two amino acid chains that are connected by disulfide linkages. When the two amino acid chains are split apart, the functional activity of insulin molecule is lost. So when you split or when you break down the disulfide linkages, the two polypeptide chains, they will lose their functional activity, so it can't stimulate the cells. Insulin is synthesized in beta cells to form initially a pre-pro-insulin. So there will be formation of pre-pro-insulin within the rough endoplasmic reticulum, and then this can be converted into pro-insulin within the rough endoplasmic reticulum. It will be transported to the Golgi apparatus where the pro-insulin will be converted into the active form of insulin, then stored within the vesicles, waiting for the signal for exocytosis. So this is the production of insulin. This initial pre-pro hormone, with the molecular weight of about 11,500 kilodaltons, is cleaved in the endoplasmic reticulum to form the pro-insulin with a molecular weight of about 9,000 kilodaltons. The pro-insulin now will be packaged into vesicles and transported to the Golgi apparatus. Most of the pro-insulin is further cleaved in the Golgi apparatus to form insulin. This is the modification that is taking place within the Golgi apparatus. By now you understand the function of cell organelles. So the Golgi apparatus is involved in the modification of proteins. This is one of the modifications that is taking place there. The conversion of pro-insulin into insulin and the peptide fragments before being packaged into the secretory granules. So now it will be packaged into the secretory granules, just waiting for the signal for exocytosis. This is the sequence of production of insulin. So we say that it's being produced as pre-pro-insulin in the rough endoplasmic reticulum. The message still is coming from the transcription of DNA into messenger RNA that will move to the ribosomes. Remember, the ribosomes, they have the 60S, 40S. At the center, we have the transfer RNA that will translate the message for the production of the pre-pro-insulin, which is a protein. Then it has got some segments of amino acids that are not required for the active hormone. So those are inactivating the insulin. So this structure of insulin is called pre-pro-insulin. Then there will be cleavage of the signal sequence. So you can see the cleavage of the signal sequence that will convert the pre-pro-insulin into pro-insulin. Then there will be other enzymes that will remove the C-peptide. So you can see the C-peptide here that is cleaved off as well to convert the pro-insulin into the mature insulin. Now you have the two polypeptide chains that are connected by disulfide bonds. So you can see the disulfide bonds that are connecting the two polypeptide chains. That's why we are saying that insulin is composed of two polypeptide chains, one alpha chain and one beta chain. This is the normal structure of insulin that can go and bind to the insulin receptors and trigger cellular response. And the different types of cellular response we are going to discuss as we proceed. So even that, this diagram is just summarizing the biosynthesis of insulin. To some extent, I've already explained. So you have the nucleus, there is transcription of messenger RNA into a messenger RNA. The messenger RNA will contain information on how the ribosomes are going to synthesize the pre-pro-insulin. So it will go to the ribosomes and then they will synthesize a protein. There is translation taking place there. Then you have the synthesis of pre-pro-insulin that will be packaged into vesicles, transported by molecular motors to the Golgi apparatus. In the Golgi apparatus, you have modification of pre-pro-insulin that will be converted into pro-insulin. The pro-insulin now will be packaged into vesicles, then to be stored as insulin, because now there is cleavage of the C-peptide, and you have a lot of these vesicles that will contain the active form of insulin, just waiting for the signal for exocytosis. So these vesicles now, for them to be exocytosed by the beta cells, it requires a stimulus. The stimulus here is an increase in blood glucose levels. That's why an increase in blood glucose levels will cause exocytosis of insulin. The mechanism that we'll be explaining in just a moment. When insulin is secreted into the blood, it circulates almost entirely in unbound form, meaning that it's not binding to any plasma proteins. It has a plasma half-life that averages only about 6 minutes. So when insulin is released, it's only going to survive about 6 minutes in its functional form. So the half-life of insulin is 6 minutes, so that it is mainly cleared from the circulation within 10 to 15 minutes. So within 10 to 15 minutes, you won't find the active insulin after it has been secreted. The half-life is 6 minutes, then 10 to 15 minutes, it will be cleared off from the circulation. Except for that portion of the insulin that combines with the receptor in the target cells. The remainder is degraded by the enzyme insulinase. The insulinase is the enzyme that will break down insulin. Insulinase is an enzyme that is found in the liver and to a lesser extent in the kidneys and muscles. They have this enzyme that will break down insulin. So insulinase is mainly found in the liver and also the kidneys and muscles. And slightly in most other tissues, you can also find insulinase that is responsible to break down insulin. This rapid removal from plasma is important for rapid control of metabolic function, because as long as you are having this insulin, it can continuously stimulate the cells to internalize amino acids, glucose, free fatty acids. So you need not necessarily free fatty acids, but amino acids and also glucose. So you need the insulinase enzyme to break down the insulin once it has done its function. This is a human insulin molecule. You can see it. You have two chains of amino acids, the beta chain and the alpha chain of amino acids. They are connected by disulfide bonds. The mechanism of insulin secretion that is taking place here. So you really need to understand this mechanism, because I like such questions. I can bring an essay to say, can you explain how the beta cells are going to release insulin, the production and secretion of insulin? So you need to know all that pathways. So now let's look at the mechanism by which the beta cells or the pancreatic islets of Langerhans are going to secrete insulin. So the first thing that you need to know are the receptors and the channels. So remember, I told you to say that the insulin is stored within vesicles, just waiting for the signal for the exocytosis. So there are channels that are located in the plasma membrane of the beta cells. These are called GLUT2 channels. So it's very important for you to differentiate these channels, because the GLUT2 channels are always open. They are not going to close or open, or they are not going to be translocated into the plasma membrane and later on internalized like the GLUT4 channels that are found in muscle cells. Here you have the GLUT2 channels which are always open. The other cells that will have GLUT2 channels are the nerve cells, especially the brain cells, the neurons. They have a lot of GLUT2 channels, so they don't depend on insulin for them to internalize glucose. The brain cells don't depend on insulin for them to internalize glucose, because the brain cells, they are only using glucose as a form of molecules for utilization of energy. It's only glucose that will be converted into energy, like ATP, for metabolic processes of the brain cells. So the neuronal cells, they do have a lot of GLUT2 channels. The pancreatic cells, the beta cells or the pancreas, they also have GLUT2 channels which are always open. They are always open, so glucose will start moving down the concentration gradient. After a meal, the blood glucose level is going to increase. Then you have a higher concentration of glucose in the interstitium that will start diffusing into the beta cells. So this is facilitated diffusion that is taking place here. So they do have a lot of GLUT2 channels. Glucose will move into the beta cells. This glucose will be worked on by an enzyme, the glucokinase enzyme, that will cause a phosphorylation reaction. I mean, it's a kinase. What does it do? It catalyzes a phosphorylation reaction. So it's going to add a phosphate to the glucose. So you can see the glucose 6-phosphate. So it means on carbon number 6, there is a phosphorylation that is taking place there to convert glucose into glucose 6-phosphate. Then you have oxidation of glucose 6-phosphate Then after oxidation, to say that this glucose will undergo glycolysis that will generate a lot of ATP. The glycolysis process itself, it produces net production of 2 ATP. So there will be an increase in the ATP concentration within the beta cells. This increase in the ATP is required because the plasma membrane or the beta cells, they also do have the ATP dependent potassium channels. ATP dependent potassium channels. In the presence of ATP, they are going to close. When they are closing, it means that potassium is not leaking out of the beta cells, it's trapped within the beta cells. So the the potassium that is trapped within the beta cells, it will cause depolarization of the membrane because you have so much of this potassium that is trapped inside of the cell. The potassium can't leak out of the cell, but you can have some potassium that is leaking into the cell, and that can cause depolarization of the membrane because the cell is gaining positively charged ions. So that will bring about depolarization of the membrane. Once the membrane is depolarizing, it will now activate the opening of calcium-voted gated ion channels. These calcium-voted gated ion channels, they are sensitive to the wave of depolarization. So there is a threshold at which they are going to open. So once you have depolarization, they are going to open. That will bring about influx of calcium. Calcium will start moving from the outside into the cells. You have calcium influx into the cells. This calcium influx, it will cause this calcium to go and bind to V snare and T snare. It will facilitate exocytosis. So you can see that the insulin that was stored within the vesicles, it will be now exocytosed to the outside. This is the release of calcium to the outside. So this is the actual mechanism by which insulin will be released to the outside of the beta cells. So I hope you'll be able to explain that. If you don't understand it, let me explain using the notes. So I have the notes here that will explain the process by which insulin is going to be released, but otherwise it's the same process. The beta cells have a larger number of glucose transporters. These are GLUT2 channels that will permit a rate of glucose influx that is proportional to the blood concentration in physiological range. So after a meal, you have an increase in the concentration of blood glucose. This blood glucose now will start entering the beta cells via the glucose transporter, the GLUT2 channels. Once inside the cell, the glucose is phosphorylated to form glucose-6-phosphate by glucokinase. This step appears to be the rate limiting for glucose metabolism in the beta cells and is considered the major mechanism for glucose sensing and adjustment of the amount of the secreted insulin to the blood glucose levels. The glucose-6-phosphate is subsequently oxidized to form adenosine triphosphate, which inhibits the ATP sensitive potassium channels. So these are the ATP sensitive or ATP dependent potassium channels I was talking about. So they will close. The closure of the potassium channels depolarizes the cell membrane, thereby opening voltage gated calcium channels, which are sensitive to changes in the membrane potential. So they are responding to a particular voltage. So after depolarization, they are going to open to allow influx of calcium. This produces an influx of calcium that stimulates the fusion of the docked insulin containing vesicles with the cell membrane and secretion of insulin into the extracellular fluid by exocytosis. So the calcium is going to enhance exocytosis of insulin because now the insulin containing vesicles, they will be docked to the cell membrane, then there will be dissolution that will release the insulin to the outside of the cells. Okay, so it's the same process here. So the first thing, you need glucose to enter the beta cells, it will be worked on by glucokinase to convert it to glucose 6-phosphate. It will undergo glycolysis for the production of pyruvate. This process will produce ATP, and this pyruvate can enter the mitochondria in the electron transport chain to produce more ATP and ADP. If you have an increase in ATP, it will cause closure of ATP sensitive potassium channels, blocking the leakage of potassium. An increase in the potassium concentration will cause depolarization, opening the voltage dependent calcium channels. Then calcium influx. Calcium is moving from the outside into the cell because you have more calcium in the outside as compared to the inside of the cell. The concentration inside is almost 0.001, but the concentration outside is 2.4 to about 2.6. This calcium that is entering the cell, it will facilitate exocytosis by binding to the V snare, T snare. Then you have exocytosis of insulin that will be released. Then you have other hormones and other ligands that can also work via the G protein coupled receptors. So you can see the incretins, the G protein coupled receptors that can increase the cyclic AMP, and this can also bring about exocytosis. So there are certain hormones that can increase exocytosis of insulin. So you will see the relationship of other hormones that can increase the secretion of insulin. At the same time, you have sulfonylurea drugs that can also inhibit the function of ATP sensitive potassium channels that can increase the potassium within the cell. It can also bring about depolarization and increasing the secretion of insulin. So there are sulfonylurea drugs that can be used in diabetes type 2, especially, that can increase the release of insulin. So this insulin can go and bind to certain receptors that are still functional. Because you know, to say in type 2 diabetes, it's a resistant issue whereby the receptors are resistant to insulin, but there are still some receptors which are still functional. So those receptors that are still functional, if you increase the concentration of insulin, you can also increase chances of interacting with those functional receptors for insulin. Then the cells can respond in diabetes. So treatment or management of diabetes type 2, you can use sulfonylurea drugs that can inhibit the ATP sensitive potassium channels. Okay, we make progress. Other nutrients such as certain amino acids can also be metabolized by the beta cells to increase intracellular ATP levels and stimulate the insulin secretion. So other nutrients such as amino acids, they can also increase the secretion of insulin by increasing the intracellular ATP. They can be used to be converted into ATP. Then that can also increase the secretion of insulin because the ATP will now be blocking the ATP sensitive channels for potassium. Some hormones such as glucagon, gastric inhibitory peptide, as well as acetylcholine will increase the intracellular calcium levels through other signaling pathways, enhance the effect of glucose, although they do not have major effect on the insulin secretion in the absence of glucose. So in the presence of glucose, some of these hormones will also enhance the secretion of insulin, like glucagon, gastric inhibitory peptide, and acetylcholine. But you realize to say glucagon is inhibited when you have a lot of insulin. So that in itself, it can regulate the secretion of glucagon if you have excessive amount of insulin that is being secreted there. Other hormones including somatostatin, norepinephrine, by activating the alpha-adrenergic receptors, they will inhibit exocytosis of insulin. So here you can see how somatostatin and norepinephrine is going to inhibit the secretion of insulin. The sulfonylurea drugs will stimulate insulin secretin by binding to ATP sensitive potassium channels and blocking their activity. So they are blocking the activity of ATP sensitive potassium channels. So there will be no movement of potassium. It will be trapped within the cell. That will bring about depolarization, increasing the depolarization and then opening the calcium voltage gated ion channels. The influx of calcium will bring about exocytosis of insulin. So you are going to increase the secretion of insulin. And these are some of the drugs that can be used in type 2 diabetes, which I said. This results in depolarizing effect that triggers insulin secretion, making these drugs very useful in stimulating insulin secretion in patients with type 2 diabetes, which makes sense. The control of insulin secretion, what will be controlling the insulin secretion? The increased blood glucose levels. So if you have an increase in blood glucose levels are above the normal acceptable range, so above the normal acceptable range, if the blood glucose levels is increasing greater than 100 milligrams per deciliter, that will stimulate insulin secretion, because much of this glucose will be entering the beta cells via the GLUT2 channels. They need to be worked on by glucokinase to be converted into glucose 6-phosphates that will undergo oxidation, glycolysis, production of ATP. Then it can also, the pyruvate can enter the mitochondria to join the electron transport chain. That will produce a lot of ATP in the electron transport chain. You know, to say that the movement of ions, the movement of electrons is coupled with the production of ATP. So it's called oxidative phosphorylation in the mitochondria. There will be an increase in ATP. Then that will be blocking now the ATP sensitive potassium channels, depolarizing the membrane, opening the calcium voltage gated ion channels, calcium entering, that will facilitate exocytosis. Then you have secretion of insulin. So an increase in glucose level above the normal acceptable ranges will stimulate insulin secretion. Some of the amino acids will stimulate insulin secretion as well. Lysine, arginine. Lysine and arginine are the most potent that can also stimulate the release of insulin, because for them to be internalized by other cells, they also need insulin to go and open up those cells. So you find that insulin will go and bind to those cells to introduce channels that can internalize the amino acids. So you know, to say that the presence of these amino acids can also stimulate the secretion of insulin, which makes sense. They strongly potentiate the glucose stimulus for insulin secretion. Insulin in turn will promote transport of amino acids into the tissue cells as well as intracellular formation of protein. So you need the insulin to have protein metabolism, or the formation of protein in the cells, and also transportation, internalization of amino acids by the cells also requires the effect of insulin, just like the internalization of glucose by the muscle cells, adipose cells, and also hepatocytes. Gastrointestinal hormones, a mixture of several important gastrointestinal hormones which we discussed, like gastrin, secretin, cholecystokinin, gastric inhibitory peptide, which seems to be the most potent, causes a moderate increase in insulin secretion, because most of these hormones are being secreted after a meal. So you know, to say they are going to increase the secretion from the gastric pits, they are going to increase intestinal secretions, they are going to increase secretions from the gallbladder, they are going to increase the secretions from the pancreas. So because they are increasing the secretions for the GIT digestion and absorption, you also know, to say that these hormones, they can also increase the secretion of insulin. So these hormones are released in gastrointestinal tract after a person eats a meal. That's what I said. After then cause an anticipatory increase in blood insulin in preparation for the glucose and amino acids that has to be absorbed from the meal. So you know, to say the body will start preparing itself, because it will anticipate that the blood glucose levels is going to increase, amino acids levels are going to increase. So you need to open up the cells So the key to open up the cells to internalize these minerals or these nutrients, you need insulin. So insulin is the one that will be secreted. So most of these hormones can also stimulate the secretion of insulin, which makes sense. These gastrointestinal hormones generally act the way as amino acids to increase the sensitivity of insulin response, to increase blood glucose. So you know to say when you have an increase in blood glucose, the amino acids, we said, is going to enhance secretion of glucose, and also these hormones can also increase the secretion of insulin. Other hormones and autonomic nervous system. Other hormones in the autonomic nervous system that can also stimulate or inhibit the secretion of insulin. Other hormones that either directly increase insulin secretion or potentiate the glucose stimulus for insulin secretion will include glucagon, growth hormone, cortisol, and to a lesser extent progesterone and estrogens. So now you know to say glucagon can stimulate insulin secretion. The growth hormone can also stimulate insulin secretion, because we said growth hormone is it has got anti-insulin factor. So because it has got anti-insulin factor, then it's inhibiting the cells to internalize glucose. An increase in glucose levels, that in itself will go and increase insulin secretion. So you can see here that the growth hormone can also increase insulin secretion. The cortisol levels, if you have an increase in cortisol levels, it can also increase insulin secretion, because cortisol is going to stimulate gluconeogenesis. Then it's also going to increase blood glucose levels, because it's encouraging gluconeogenesis, and that can also cause an increase in insulin. So it's somehow related to the glucose levels. Progesterone and estrogens, to some extent, they can also cause an increase. The importance of stimulatory effect of these hormones is that prolonged secretion of any of them in large quantities can occasionally lead to exhaustion of the beta cells or the islets or Langerhans, thereby increasing the risk for developing diabetes mellitus. So some of the risk factors in the development of diabetes mellitus is the secretion of these hormones in higher concentration for a long period of time, because these hormones, we are saying that they can increase the secretion of insulin. So they are overstimulating the beta cells. The overstimulation of the beta cells, then the beta cells, they can undergo degeneration that can result into development of diabetes mellitus. Indeed, diabetes often occurs in people who are maintained on higher pharmacological doses of some of these hormones. So if you're using much of cortisol, you're using much of steroid hormones, all those, they can cause an increase in insulin levels, and then that can result into diabetes. So they can cause diabetes. The beta cells will degenerate, and then you can have type 1 diabetes. And sometimes you'll find that there will be resistance by the receptors, the insulin receptors, and then you can have type 2 diabetes. Diabetes is particularly common in giants or acromegalic people. So in acromegalic people and giantism, we discussed this. I told you to say giants is due to the production of growth hormone, and the growth hormone has anti-insulin effect on cells. So it means that the cells won't be actually responding to insulin, because that the effect of insulin on the cells will be inhibited by the growth hormone. So that in itself will result into an increase in glucose levels, and that glucose levels can overstimulate the production of insulin by the beta cells, and the beta cells undergo degeneration. That will also result into a full-blown picture of diabetes mellitus. So diabetes is particularly common in giants or acromegalic people with growth hormone secreting tumors, or in people whose adrenal glands secretes excessive glucocorticoids. So even excessive amount of glucocorticoids like cortisol can also result into adrenal diabetes. If you remember, excessive amount of cortisol, continuous production of cortisol, or pharmacological doses of cortisol, it can cause adrenal diabetes. So adrenal diabetes mainly is not being caused by pharmacological doses of cortisol, but the overproduction of cortisol by the adrenal gland in hyperadrenalism. So we said in hyperadrenalism, or Cushing's syndrome or Cushing's disease, whereby the adrenal cortex is overproducing cortisol, that can cause adrenal diabetes. The opposite can cause adrenal crisis. Okay, I hope you can still remember that. Under the same conditions, stimulation of parasympathetic nerves to the pancreas can increase insulin secretion. So the sympathetics will inhibit, the parasympathetics can increase insulin secretion under certain conditions. This table is summarizing conditions that can increase or decrease insulin secretion, or the factors that can increase or decrease insulin secretion. So the factors or condition that can increase insulin secretion, the first one is increased blood glucose, the mechanism which we've already discussed. Increased blood free fatty acids. Free fatty acids can also increase the amount of insulin that is being secreted. Increased amino acids also can increase secretion of insulin. Gastrointestinal hormones, which I've also explained, the gastrin, cholecystokinin, secretin, gastric inhibitory peptide, is going to cause anticipatory increase in insulin secretion after a meal, especially. Glucagon, growth hormone, cortisol, all these can cause an increase in insulin secretion. The parasympathetic stimulation, acetylcholine, is going to increase. The beta-adrenergic stimulation, the insulin resistance itself in obesity. The insulin resistance is because the cells are not responding to insulin. So the beta cells will think that maybe they are not producing enough insulin, so they'll overproduce insulin. So you find that insulin resistance will result into overproduction of insulin. That takes place in obesity because the cholesterol, the free fatty acids, they can interfere the interaction between the receptors and the insulin itself. Sulfonylurea, the sulfonylurea drugs like glyburide, the glyburide or 2-butamide, all these drugs, they can increase the insulin secretion because they are inhibiting the ATP sensitive potassium channels. If you remember, the sulfonylurea drugs. Then the factors that are decreasing insulin secretion, we have quite a number of them as well. Decreased blood glucose is the opposite. Fasting is the opposite. So you're talking of hypoglycemia. Hypoglycemia is going to inhibit the secretion of insulin. Somatostatin is going to inhibit the secretion of insulin. Then you have the alpha-adrenergic activity. This is the sympathetic stimulation, is going to inhibit insulin secretion because the GIT is activated by the parasympathetics. That's why the parasympathetics is going to increase insulin secretion. Then the sympathetics is going to inhibit the secretion of insulin. Leptin is a hormone that is produced by adipose tissue and is being produced when somebody is obese. So you inhibit the production of insulin because you are already obese. You don't want to accumulate more fat or accumulate more protein, so it's going to inhibit the secretion of insulin. Then it's the same hormone that is inhibiting the hunger center and is stimulating the satiety center in the hypothalamus. So I hope you remember that. Activation of target cell receptor by insulin and resulting in cellular effect. What's the action of insulin, or the mode of action for insulin? How is it going to activate the receptor? So you need to remember the structure of the insulin hormone, then how it's going to interact with the insulin receptors, activating the insulin receptor substrate, activating the tyrosine kinase. So you need to understand. Such a question can also come as an essay question. To initiate its effect on target cell, insulin first binds with and activates a membrane receptor protein. So it's all about receptor. So here there are specific receptors for insulin. Remember, they are going to stimulate the target cells by binding to specific receptors. It is the activated receptor, not the insulin, that causes subsequent effect within the cells. The insulin receptor is a combination of 4 subunits held together by disulfide linkages, 2 alpha subunits and 2 beta subunits. So we have 2 alpha protein subunits and 2 beta protein subunits. The 2 alpha protein subunits, this is a structure of the receptor. The 2 alpha protein subunits lie entirely outside of the cell membrane. So it's outside the cell membrane, it's outside. The 2 beta protein subunits that penetrate through the membrane, protruding into the cell cytoplasm. So this is the one that will be linked to an enzyme. So it's an integral protein. Then the 2 alpha protein subunits, they are more like peripheral protein that are just attaching to the 2 beta protein subunits. That is crossing the plasma membrane and protruding into the cytoplasm. That will be activating the enzyme once insulin goes and binds. The insulin binds with the alpha subunits on the outside of the cell. Remember, insulin is fat soluble. No, it's water soluble. Insulin is water soluble. It's a protein. So if it's a protein, it's water soluble. It can't actually dissolve in plasma. It will dissolve in plasma because it's a protein. I mean, it's a protein. It's water soluble. It will dissolve in plasma, but it doesn't dissolve in fat. So it doesn't cross the phospholipid bilayer to enter the cells. So it's going to bind to receptors at the level of the cell membrane. Okay, I hope we are on the same page. So the insulin binds with the alpha subunits on the outside of the cell, but because of the linkages with the beta protein subunits, the portions of the beta subunits protruding into the cell become autophosphorylated. So they will become autophosphorylated when there is interaction between the receptor and the alpha protein subunits. Thus, the insulin receptor is an example of enzyme-linked receptor. So remember, when we are discussing the general mechanism by which hormones are going to activate the cells, or the signaling pathways, the intracellular signaling pathways, we had a number of such channels. We had ligand-gated ion channels, we had enzyme-linked channels, we had also G-protein coupled ion receptors, G-protein coupled receptors, if you remember that. So an insulin receptor is an example of enzyme-linked receptor. So it's a receptor that is linked to an enzyme. Once the insulin goes and binds, it will activate the enzyme, which is the tyrosine kinase. Autophosphorylation of the beta subunits or the receptor activates the local tyrosine kinase, which in turn causes phosphorylation of multiple other intracellular enzymes, including a group of insulin receptor substrate. So the IRS, the insulin receptor substrates, will be activated by the tyrosine kinase after autophosphorylation has taken place. Different types of insulin receptor substrate, insulin receptor substrate 1, 2, 3, and so on, are expressed in different tissues. The net effect is to activate some of these enzymes while inactivating others. So some of the enzymes are activated, other enzymes are inactivated in the process. In this way, insulin directs the intracellular metabolic machinery to produce the desirable effects on carbohydrate, fat, protein metabolism. So sometimes the notes is very complicated for you to understand, but from a diagram you'll be able to pick up the summary of what is happening here. So we have insulin that is composed of 2 polypeptide chain. So one polypeptide chain will bind to alpha protein subunit and the other one will bind to another alpha protein subunits. So you have 2 pairs of alpha protein subunits and 2 pairs of beta protein subunits that are combined to form the insulin receptor. The binding sites are found on the alpha protein subunits that are connected by disulfide bond, just like the insulin 2 alpha and beta chains, they are connected by the disulfide bonds. So those two parts, they will come and bind to the alpha protein subunits. That will activate the beta part of the receptor that is linked to tyrosine kinase. That will cause autophosphorylation of tyrosine kinase. The insulin receptor substrate will be phosphorylated as well. So the tyrosine kinase is a kinase that will start phosphorylating other enzymes. So they will activate other enzymes, which are called insulin receptor substrate phosphorylation of enzymes. So there are a number of enzymes that are being activated. Some of these enzymes, they're involved in glucose transport that will cause translocation of GLUT4 channels in a muscle cell or adipose tissue. We have the GLUT4 channels that will be translocated and embedded within the plasma membrane. That will allow this particular cell now to internalize glucose. Without insulin, this GLUT4 channel won't be there. The glucose will be trapped outside of the cell. But for this cell to open up, it requires insulin to come and bind, activating the beta, the tyrosine kinase that will cause autophosphorylation. Then once the tyrosine kinase is autophosphorylated, it will start activating the insulin receptor substrate, phosphorylating other enzymes that will cause translocation of GLUT4 channels to be embedded within the plasma membrane of the cell. Then it will open up to allow influx of calcium. So calcium will be moving from the interstitium into the cells. It could be the muscle cell or adipose cell, or it could be hepatocyte. Then you also have activation of other enzymes that are involved in protein synthesis. So you have protein synthesis. This protein, they could be GLUT4 channels that are being produced now that will be embedded later on. So you have immediate effect of insulin and long-term effect of insulin. So this could be one of the long-term effect of insulin that will bring about protein synthesis. It will bring about fatty synthesis. It will bring about glucose synthesis like gluconeogenesis enzymes. Then it will also bring about growth and gene expression, which are also long-term effect of insulin. So we have the short-term or the immediate effect of insulin. Then we also have long-term effect of insulin. But insulin, the half-life of insulin is 6 minutes. It will take about 10 to 15 minutes to metabolize the insulin, and then their effect can remain in action after the insulin has been destroyed or metabolized by the liver and the other tissues that contain insulinase enzyme. Okay, so it's the same information here. We have insulin molecule that will come and bind to insulin receptor. It's composed of 2 alpha and 2 beta chains. So it will bind to the alpha chains, activating the beta chains that is linked to an enzyme, the tyrosine kinase, that will now phosphorylate other enzymes, activating the insulin signal pathway. It will have an effect on the lipid metabolism, effect on the body growth or cell growth. It will have an effect on protein metabolism. Then it can cause translocation of GLUT4 vesicles. So in these cells, they have GLUT4 containing vesicles in the cytoplasm. In the absence of insulin, the GLUT4 will be found within the membrane of the vesicles, but with the presence of insulin, they will be translocated. Then they will be embedded within the plasma membrane of these particular cells. Then the cells are opening up to allow movement of glucose. Glucose will start entering the cell. That's how it works. Let's look at the effect of insulin stimulation and the end effects of insulin stimulation are as follows. So what are the end effects of insulin stimulation? Within seconds after insulin binds with the membrane receptor, the membranes of about 80% of the body cells might increase their uptake of glucose, which makes sense because now they will have GLUT4 channels that will be used for internalization of glucose. This is essentially true of muscle cells, adipose cells, but it's not true of most neurons in the brain, because these neurons, they don't necessarily depend on insulin for them to internalize glucose. They depend on glucose as a source of energy. So you find that whether you have insulin, you don't have insulin, they will still internalize glucose. The increased glucose transported into the cells is immediately phosphorylated and becomes a substrate for all the usual carbohydrate metabolic functions. The increased glucose transport is believed to result from translocation of multiple intracellular vesicles to the cell membrane. These vesicles carry in their own membrane multiple molecules of glucose transporter proteins, which are the GLUT4 transporter proteins, which bind with the cell membrane and facilitate glucose uptake into the cells. When insulin is no longer available, these vesicles separate from the cell membrane within about 3 to 5 minutes and move back to the cell interior to be reused again and again as needed. So once you don't have insulin, there will be internalization of these GLUT4 channels. So they will go back. There will be invagination and internalization of the GLUT4 channels into the vesicles, back into the cells. Then if you have another insulin, then they'll be translocated. So they can be reused again and again. That's what it means. The cell membrane becomes more permeable to many of amino acids, potassium ions, and phosphate ions, causing increased transport of these substances into the cell. The slower effects. So we have the slower effects and the much slower effects of insulin. The slower effects occur during the next 10 to 15 minutes to change the activity level of many more intracellular metabolic enzymes. These effects result mainly from the changed state of phosphorylation of the enzymes. So once those enzymes are phosphorylated, they can continue having an effect on the cell. The much slower effects continue to occur for hours and even several days. They result from changed rates of translation of messenger RNAs at the ribosomes to form new proteins, and still slower effects from changed rates of transcription of DNA in the cell nucleus. So there could be some transcription of DNA into messenger RNA, the proteins that will later on become GLUT4 channels or other enzymes that are involved in fat metabolism, lipid metabolism, mention them. The effect of insulin on carbohydrate metabolism. So now we are going to look at the effect of insulin on all macromolecules, carbohydrate, lipids, and proteins. The effect of insulin on carbohydrate metabolism. What is the effect? Immediately after a high carbohydrate meal, the glucose that is absorbed into the blood causes rapid secretion of insulin. The insulin in turn causes rapid uptake, storage, and use of glucose by almost all tissues of the body, but especially by the muscles, adipose tissue, and the liver cells. Not the neurons, especially the neurons of the central nervous system. They don't depend on glucose at all. They don't depend on insulin at all, but they depend on glucose as a source of energy. Insulin is going to promote muscle glucose uptake and metabolism. Of course, by the introduction of GLUT4 channels, internalization of glucose, and metabolism of this glucose inside the cell. During much of the day, muscle tissue depends not on glucose for energy, but on fatty acids. So you need to take note. So when you are resting, the muscle cells are using much of fatty acids as a source of energy, not glucose. Why? It's because that is way after your meal, and then the insulin levels are low. So these cells, they can't internalize glucose because they don't have much insulin. So they'll be depending on fatty acids. Fatty acids, they can cross a phospholipid bilayer. They don't depend on insulin. That's why you find that muscle cells, when you're just resting, they'll be using much of free fatty acids as a source of energy. The principal reason for this is that the normal resting muscle membrane is only slightly permeable to glucose, except when the muscle fiber is stimulated by insulin. So if you don't have insulin, you can't internalize glucose, so you'll be using free fatty acids. Between meals, the amount of insulin that is secreted is too small to promote significant amount of glucose entry into the cells. However, under two conditions, the muscles do use large amounts of glucose. One of these is during moderate or heavy exercise. During moderate and heavy exercise, you can be using a lot of glucose, even in absence of insulin, because exercise itself, it will increase the permeability of the membrane to that of glucose, even in absence of insulin. This usage of glucose does not require large amounts of insulin because exercising muscle fiber become more permeable to glucose even in the absence of insulin because of the contraction process itself that will cause the muscle fiber to become more permeable to glucose in absence or in smaller quantities of insulin. The second condition for muscle usage of large amounts of glucose is during the few hours after a meal. After this time, the blood glucose concentration is high and the pancreas is secreting larger quantities of insulin. The extra insulin causes rapid transport of glucose into the muscle cells. This causes the muscle cell during this period to use glucose preferably over the fatty acids. So you'll be using much of the glucose rather than the fatty acids, because now it's after a meal, you are producing a lot of insulin or you are secreting a lot of insulin. The cells can afford to internalize glucose now because they are sensing the presence of insulin that is binding to the insulin receptors, translocating GLUT4 channels, opening up the cells to internalize glucose. So they'll start using much of glucose as a source of energy. That is immediately after a meal. Okay. Storage of glycogen in muscle. If the muscles are not exercising after a meal, yet glucose is transported into the muscle cells in abundance, then most of the glucose is stored in form of muscle glycogen instead of being used for energy. So now, after a meal, you are not exercising, the muscle cells, they are internalizing a lot of glucose, they're using part of that glucose as a source of energy, but there is still an overflow of glucose in the cells. So that glucose now will be stored in form of glycogen. So the glycogen can later be used for energy by the muscles. So the muscle cells, they will preserve the glycogen by converting it into, they will preserve the glucose by converting it into glycogen. Then when they need the glucose back, then there will be breakdown of glycogen in the process of glycolysis or glycogenolysis actually. It is especially useful for short periods of extreme energy use by the muscles and to provide spurts of anaerobic energy. So remember the glycogen lactic acid system that can provide energy for muscle activities. This is what it's pointing at. Insulin promotes liver uptake, storage, and use of glucose. So the insulin itself is going to promote liver uptake of glucose, storage of glucose in form of glycogen in the liver, and also use of glucose that can be used to convert it into amino acids and fats to some extent. Insulin causes most of glucose absorbed after a meal to be stored almost immediately in the liver in the form of glycogen, just like in muscle cells. Then between meals, when food is not available, the blood glucose concentration begins to fall, insulin secretion decreases rapidly, and the liver glycogen is split back into glucose, which is released back into the blood to keep the glucose concentration from falling too low. So you can see the function of the liver here is going to resist change in blood glucose levels. So it's involved in the regulation of blood glucose levels. If there is a lot of glucose in the blood, it will take up that glucose and convert it into glycogen stored within the hepatocytes. When there is hypoglycemia, there is low blood glucose levels during fasting, when you're not eating anything, then the liver will break down the glycogen that is stored within the liver in the process of glycogenolysis. Then the glucose will be released back into the blood, trying to resist change in the concentration of glucose in the blood. The mechanism by which insulin causes glucose uptake and storage in the liver includes several almost simultaneous steps that are listed below there. So they will take place almost at the same time. Insulin inactivates liver phosphorylase. The liver phosphorylase is a principal enzyme that causes liver glycogen to split into glucose. So it's going to inhibit it so that much of the glycogen is not being broken down into the glucose. Insulin causes enhanced uptake of glucose from the blood by the liver cells. It does this by increasing the activity of enzyme glucokinase, which is one of the enzymes that causes the initial phosphorylation of glucose after it diffuses into the liver cells. And then it will be stored in form of glycogen. Okay, that will initiate a pathways of metabolism that are involved in fat metabolism, glucose metabolism, and other protein metabolism. Insulin also increases the activity of enzymes that will promote glycogen synthesis, including especially the glycogen synthase. So there will be an increase in the production of glycogen synthase that is involved in glycogenesis, meaning that the conversion of glucose into glycogen. Okay, so that will be encouraged by the presence of glycogen synthase. It will increase the production and the enzyme itself, the activities of glycogen synthase. The net effect of all these actions is to increase the amount of glycogen in the liver. The glycogen can increase to a total of about 5 to 6% of the liver mass, which is equivalent to almost 100 grams of stored glycogen in the whole liver. So you have much of the glycogen that is stored there. Glucose is released from the liver between meals, which you know now. Between meals, the glycogen that is stored within the hepatocytes will be released into the blood to try and resist change in blood glucose levels so that you maintain the normal range of blood glucose levels. When the blood glucose level begins to fall to a low level between meals, several events transpire that cause the liver to release glucose back into the circulating blood. So what are those things? The decreasing blood glucose causes the pancreas to decrease its insulin secretion. So insulin secretion is going to decrease. The lack of insulin then reverses all the effects listed earlier for glycogen storage, essentially stopping further synthesis of glycogen in the liver and preventing further uptake of glucose by the liver cells from the blood. So it will be prevented so that the blood glucose increases in the blood. Lack of insulin again, along with increase in glucagon, which is discussed later. So the other lecture will be discussing glucagon. So the lack of insulin along with the increase in glucagon activates the enzyme phosphorylase. So now the phosphorylase is being activated. That will break down glycogen, or it will split glycogen into glucose phosphates. Then the glucose phosphates will be converted into glucose. So now you're breaking down glycogen, glycogenolysis. The enzyme glucose-6-phosphatase, which had been inhibited by insulin, now become activated by the insulin lack and causes the phosphate radicals to split away from the glucose. This allows the free glucose to diffuse back into the blood. So the glucose 6-phosphates, it can't diffuse because the phosphates, they are negatively charged, so it can't cross the phospholipid bilayer. So you find that most of it will be converted to glucose, and then it can move via certain channels for it to move back into the blood. If it's in the liver cells, of course, it's also using the GLUT2 channels. Hepatocytes, certain hepatocytes, they have GLUT2 channels that can also allow glucose to diffuse out of the cells to go into the blood. Thus, the liver removes glucose from the blood when it is present in excess after a meal and returns it to the blood when the blood glucose concentration falls between meals. Insulin promotes conversion of excess glucose into fatty acids and inhibits gluconeogenesis in the liver. So if you have insulin, it's going to inhibit gluconeogenesis because, you already have a lot of glucose. Why do you want to start producing glucose from non-carbohydrate sources? Because the glucose is in abundance. So the insulin is going to promote the conversion of excess glucose into fatty acids and inhibit gluconeogenesis in the liver, which also makes sense. When the quantity of glucose entering the liver cells is more than can be stored as glycogen or can be used for local hepatocyte metabolism, insulin promotes the conversion of all the excess glucose into fatty acids. So now if you have more glucose, that can be converted into glycogen and that can be used for hepatocyte metabolism. The excessive amount of glucose can be converted into fatty acids, and sometimes it can also be converted into amino acids. These fatty acids are subsequently packaged as triglycerides in very low density lipoprotein. This is the production of low density lipoprotein by the liver cells. So the glucose that is converted into fatty acids will be packaged as triglycerides in very low density lipoprotein and transported in this form by way of blood to the adipose tissue and deposited as fat. So it will be transported as low density lipoprotein to the adipose tissue, then to be deposited as fat. Then you start gaining as well because there will be fat deposits. Insulin also inhibits gluconeogenesis. I've already mentioned that. It does this mainly by decreasing the quantity and the activities or the liver enzymes required for gluconeogenesis. So those enzymes that are required for gluconeogenesis will be inhibited by insulin. Also, insulin decreases the release of amino acids from the muscle and other extrahepatic tissues, and in turn, the availability of these necessary precursors required for gluconeogenesis. So it's going to inhibit mobilization of amino acids from the extrahepatic tissue to the liver cells because you already have a lot of glucose there that can be converted into amino acids, and then it's going to inhibit gluconeogenesis. So you don't need the amino acids for you to have gluconeogenesis because gluconeogenesis is inhibited. So you're not mobilizing amino acids, you're not mobilizing fat from other cells. Lack of effects of insulin on glucose uptake and usage by the brain cell. To some extent, I've already mentioned to say the brain cell don't depend on insulin for them to internalize glucose because they mainly depend on glucose as a source of energy. So whether there is insulin, there is no insulin, they just have to internalize glucose. The brain is quite different from most other tissues of the body in that insulin has little effect on uptake or use of glucose. Instead, the brain cells are permeable to glucose and can use glucose without the intermediation of insulin. The brain cells normally use only glucose for energy. Therefore, it is essential that the blood glucose level always be maintained above a certain critical level. When the blood glucose falls too low, symptoms of hypoglycemic shock develops. because these cells, they depend on glucose only as a source of energy. So the glucose has to be maintained above a certain critical level, because if it goes below a certain critical level, then a person can actually faint, because the neurons, they are not accessing enough glucose. They won't have energy for metabolism. Then you can go into a coma because of that. Effect of insulin on carbohydrate metabolism in other cells. Insulin increases glucose transport into and glucose usage by most other cells of the body, with the exception of the brain cells, of course, because they're not depending on insulin. The transport of glucose into adipose cells mainly provide substrate for the glycerol portion of the fatty acids. So even for the production of fatty acids or fat molecules, you need the glucose that will provide the backbone. The carbon backbone of the glucose molecule is called the glycerol molecule. Therefore, in this indirect way, insulin promotes deposition of fat in these cells, because it will provide the glycerol backbone of fatty acids. So the fatty acids will go and bind for the formation of triglycerides, diglycerides, and monoglycerides. Effects of insulin on fat metabolism. Insulin promotes fat synthesis and storage. So it's going to encourage fat production or synthesis of fat. Insulin has several effects that lead to fat storage in adipose tissue. First, insulin increases the utilization of glucose by most of the body tissues, which automatically decreases the utilization of fat, thus functioning as a fat sparer. So it's going to spare fat, because if you have a lot of glucose, the body now will resort to using glucose as a source of energy. This is immediately after a meal, when you have sufficient amount of insulin. So you're internalizing a lot of glucose. Some of this glucose is being converted into glycogen, and some of the glucose will be converted into fat or amino acids, and then to be deposited in adipose tissue. So you're going to spare fat as you are using glucose as a source of energy. So it will function as a fat sparer. Insulin also promotes fat acid synthesis, especially true when more carbohydrates are ingested than can be used for immediate energy, thus providing the substrate for fat synthesis. So you start producing fat. So you have the glycerol backbone that you need for the production of triglycerides, monoglycerides, and diglycerides. Almost all these syntheses occur in the liver cells, and the fat acids are then transported from the liver by way of blood lipoproteins to adipose cells to be stored. So they are not produced in the fat cells, they are still produced in the liver cells. So the conversion of glucose into fat is done within the liver cells, and then to be transported in form of lipoprotein to the adipose tissue for storage. This story you also discuss in biochemistry. So most of this information actually I'm also discussing with you biochemistry. So I hope you'll be able to open up and answer some questions in biochemistry, physiology as well. The different factors that lead to increased fatty acid synthesis in the liver will include the following. So the different factors that lead to increase in fatty acid synthesis in the liver will include the following. Insulin increases the transport of glucose into the liver cells. After the liver glycogen concentration reaches 5 to 6%, this in itself inhibits further glycogen synthesis. Then all the additional glucose entering the liver cells become available to form fat. The glucose is first split to pyruvate in the glycolytic pathway, and the pyruvate subsequently is converted to acetyl coenzyme A, the substrate from which fatty acids are synthesized. So the glucose will undergo glycolysis to be converted into pyruvate. The pyruvate will be converted into acetyl coenzyme A, and this acetyl coenzyme A will be used now for the production of fat. An excess of citrate and isocitrate ions is formed by the citric acid cycle when excess amount of glucose are being used for energy. These ions then have a direct effect in activating acetyl coenzyme A carboxylase, the enzyme required to carboxylate acetyl coenzyme A to form Hormonal coenzyme A, the Hormonal coenzyme A. So the acetyl coenzyme A is converted into Hormonal coenzyme A, the first stage of fatty acid synthesis. Most of the fatty acids are then synthesized within the liver itself and used to form triglycerides. The usual form of storage of fat or fat storage, the triglycerides. They are released from the liver cells to the blood in the form of lipoproteins. Insulin activates lipoprotein lipase in the capillary walls of adipose tissue, which split the triglycerides again into fatty acids. A requirement for them to be absorbed into the adipose tissue, where they are again converted into triglycerides. So we saw this when you're looking at absorption of fat, to say when they are transported to the cells, they will be broken down into fatty acids, and then they'll cross the phospholipid bilayer and the glycerol molecules. Then in the cells, they will regenerate the triglycerides by reacting the fatty acids with the glycerol backbone as well. What is the role of insulin in storage of fat in adipose cells? It's basically the same information here. I don't know if there's anything new. Insulin has two other essential effects that are required for fat storage in adipose cells. Insulin inhibits the action of hormone-sensitive lipase. This is the enzyme that causes hydrolysis of triglycerides already stored in the fat cells. So it's going to inhibit the utilization of fat within the adipose tissue. Therefore, the release of fatty acids from the adipose tissue into the circulating blood is inhibited. Insulin is going to promote glucose transport through the cell membrane into the fat cells in exactly the same way that it promotes glucose transport into the muscle cells. So it's also going to increase the transportation. It's going to promote the transportation of glucose through the cell membrane into the fat cells, just like the way it does in the muscle cells. So it's still binding to insulin receptors, translocating of GLUT4 channels, and then facilitating internalization of glucose into the adipose tissue now. Some of this glucose is then used to synthesize minute amount of fatty acids, but more important, it also forms large quantities of a glycerol phosphate, which supplies the glycerol that combines with the fatty acids to form triglycerides that are the storage form of fat in adipose tissue. So it will be converted into fat. Therefore, when insulin is not available, even storage of large amounts of fat acids transported from the liver in the lipoprotein is almost blocked as well. Insulin deficiency decreases use of fat for energy. So if you don't have insulin, it's going to increase usage of fat for energy, because now you don't have the glucose. The body is not, the muscle cells, they're not internalizing the glucose without insulin, so they'll start using fatty acids. So this is where the fatty acids are being used for energy. Almost all aspects of fat breakdown and use for providing energy are greatly enhanced in the absence of insulin. This occurs even normally between meals when secretion of insulin is minimal, but it becomes extreme in diabetes mellitus when secretion of insulin is almost zero. So if the secretion of insulin is almost zero, the body cells, mainly, they are using fat as a source of energy. That's why diabetic patients, sometimes they lose a lot of weight because they're not using insulin. In as much as they have higher blood glucose levels, that glucose can't enter the cells because there is no insulin to open up the cells. So the body cells now be using more of fatty acids. So you find that you start using your fat deposits, you start using your protein deposits, then you start losing weight. The resulting effects are as follows. Insulin deficiency causes lipolysis of storage fat and the release of free fatty acids. In the absence of insulin, all the effects of insulin noticed area that cause storage of fat are reversed. The most important effect is that the enzyme hormone-sensitive lipase in the fat cells becomes strongly activated. This causes hydrolysis of stored triglycerides, releasing large quantities of fatty acids and glycerol into the circulating blood. Consequently, the plasma concentration of free fatty acids begin to rise within minutes. These free fatty acids then becomes the main energy substrate used by essentially all types of tissues of the body besides the brain. So the brain will still depend on glucose. It can still internalize glucose even in absence of insulin. Insulin deficiency increases plasma cholesterol and phospholipids concentration. The excess of fatty acids in plasma also promotes liver conversion of some of the fatty acids into phospholipids and cholesterol. These two substrates, along with excess triglycerides formed at the same time in the liver, are then discharged into the blood in form of lipoprotein. Occasionally, the plasma lipoprotein increases as much as threefold in the absence of insulin, giving the high total concentration of plasma. The high lipid concentration, especially the high concentration of cholesterol, will promote the development of atherosclerosis in people with serious diabetes. So again, people with diabetes, they are going to have atherosclerosis because there is deposition of cholesterol in the blood vessels, because the body is just using much of fat as a source of energy. There is lipolysis, there is activation of lipase that is breaking down fat. You have a lot of fat in circulation. Then you have an atherosclerosis, hardening of arteries. There will be an increase in pulse wave velocity. The pulse wave velocity is going to increase. There will be an overload on the heart. Then that overload on the heart can also cause cardiovascular diseases. Excess usage of fat during insulin lack causes ketosis and acidosis. This is a mechanism that you also need to understand here, how excessive usage of fat during insulin lack can cause ketosis and acidosis. So the ketosis and acidosis is not due to the usage of proteins, but mainly to the usage of fat for the mechanisms that I'm about to explain. So pay attention. Insulin lack also causes excessive amounts of acetoacetic acid to be formed in the liver cells if you are using a lot of fat in the absence of insulin. This results from the following effect. In the absence of insulin, the carnitine transport mechanism becomes increasingly activated. In the mitochondria, the beta oxidation of fatty acids then proceeds very rapidly, releasing extreme amounts of acetyl coenzyme A. So here now we have beta oxidation of fatty acids because you don't have glucose, there is no pyruvate, there will be beta oxidation of fatty acids that will be converted into acetyl coenzyme A. A larger part of this excess acetyl coenzyme A is then condensed to form acetoacetic acid. So it's the same acetyl coenzyme A that is condensed to form acetoacetic acid, which in turn is released into the circulating blood. So once the acetyl coenzyme A is converted into acetoacetic acid, it will be released into the circulating blood. Most of this passes to the peripheral cells, where it is again converted into acetyl coenzyme A and used for energy in the usual manner. So the acetyl coenzyme A now is being used as energy, because acetoacetic acid can be converted back to acetyl coenzyme A, and then it can be used by those cells as energy, because they are not using carbohydrates, they are not using proteins at this point in time. At the same time, the absence of insulin also depresses the utilization of acetoacetic acid in the peripheral cells. So you can see again that the absence of insulin is a problem here, because it's also inhibiting these cells to use the acetoacetic acid as a form of energy. Thus, so much acetic acid is released from the liver that cannot all be metabolized by the tissues. So most of this acetoacetic acid is not being used by the tissues, because the absence of insulin is also inhibiting the peripheral tissues to use acetoacetic acid. So some of the acetoacetic acid is also converted to beta hydroxybutyric acid and also acetone. So these are the three ketone bodies that are common in diabetes. So the first one is acetoacetic acid that can be converted into beta hydroxybutyric acid and acetone. These two substances, along with acetoacetic acid, are called ketone bodies. In their presence is large quantities in the body fluids is called ketosis. If you have a higher quantity of ketone bodies in the extracellular fluid, that condition is called ketosis, accumulation of ketone bodies, which is the acetoacetic acid, the beta hydroxybutyric acid, and acetone. The acetoacetic acid and the beta hydroxybutyric acid can cause severe acidosis and coma. So you have accumulation of these acids that will cause severe acidosis, and that can cause depression of the nervous system, especially the central nervous system. Then that can cause coma, which often leads to death. So the diabetic patients, when they reach this stage, then the prognosis of this disease is very bad. The end result, they will die. So if they go into coma because of the ketone bodies, then they will die. I hope this is the last slide. We are looking at the effect of insulin on protein metabolism and on growth. I'm tired also, so I think I need to wind up. Let me just quickly look at this and we end. The effect of insulin on protein metabolism and on the growth, to some extent we've discussed this several times, so I'll be a bit fast. Insulin promotes protein synthesis and storage. During the few hours after a meal, when excess quantities of nutrients are available in the circulating blood, not only carbohydrates and fats, but proteins as well are stored in the tissues. Insulin is required for this to occur. So some of the effects of insulin are: insulin is going to stimulate transport of many of amino acids into the cells. Among the amino acids most strongly transported are valine amino acids, leucine, isoleucine, tyrosine, and phenylalanine. Thus, insulin shares the growth hormone, the capability of increasing the uptake of amino acids into the tissues. So it can function just like the growth hormone to increase the uptake of amino acids into the tissues. Insulin increases the translation of messenger RNA and genetic information, of course. Over a long period of time, insulin also increases the rate of transcription of selected DNA genetic sequences. So it's going to increase that. Insulin inhibits the catabolism of proteins. Of course, it's going to inhibit the catabolism of the proteins. In the liver, insulin depresses the rate of gluconeogenesis. So in summary of everything that we've covered, insulin is going to promote protein formation and is going to prevent the degradation of proteins. Insulin lack will cause protein depletion and increase plasma amino acids. Insulin and growth hormone interact synergistically to promote growth. The two hormones function synergistically to promote growth, each performing a specific function that is separate from that of the other. So this is just a summary of the insulin effect on the body. So you can see there are a lot of function of insulin. If a question comes to say, can you discuss the metabolic functions of insulin? Then just know to say you're in trouble, because there are a lot of information that you need to mention there. You need to mention the effect of insulin on carbohydrates, on fat, and on proteins. And of course, you need to mention how insulin is going to bind to insulin receptors. If I add to say, can you also explain the biosynthesis of insulin? You need to mention all that. So we have a lot of information. So this information is also going to help you answer certain questions in biochemistry. But you know, these courses, even if we separate them to say there's anatomy, there's physiology, there's biochemistry, science is science. We have basic sciences. We can't separate them from one another. In as much as we are separating them as courses, it's just to enable us to be able to reach out to the students. Otherwise, it will be very confusing if you're learning everything at the same time. So with this information now, later on when you go into your third year, when you start looking at pathology, microbiology, you need to apply this information. Otherwise, thank you very much for your time. This is the glucagon hormone, and we wind up with other hormones, especially reproductive physiology. Okay, so this is where we end. Thank you.